Okay, last section on the evolution of our immune system. So, vertebrate animals like ourselves are evolved from a wider pool of animals called chordates. Chordates are evolved from a wider pool of animals called deuterostomes, based on the way that they make their early embryos. And so, some examples of those would be things like starfish and sea urchins. And those then emerge from a common ancestor, which is going to be similar to things like mollusks and arthropods, which are going to be stuff like insects and crustaceans, and all the other kinds of animals. So you can actually see some of these transitions in the DNA and in the way that different animals actually go about this. So if you're actually looking on our vertebrate animal tree, down toward the bottom, we have these guys called anathans. And so these are the really primitive branches of the vertebrate tree. They don't have jaws. They're going to be something like a lamprey, for example, that's just going to have basically teeth in its throat instead of an actual working jaw. And so, yeah, these things are very primitive. The earliest chordate fossils look like them. And yeah, it's interesting. You'd say, okay, so what's their immune system look like? And yeah, let's see. So animals like us are called gnathostomes, and they have jaws. Gnatho, anytime you see gnatho, think like gnashing your teeth, like that. That's it's the same root. And so all the gnathostomes are going to have adaptive immunity based on RAG-mediated VDJ recombination. So they're going to encode RAG genes, and they're going to have VD and J regions for something, whether it's antibodies or T cells or something that has a different name but functions in roughly the same way. Yeah, and I mean an antibody is just a like a loose T cell receptor basically. They're just detachable T cell receptor like molecules. So it kind of makes sense that these things all sort of evolved from a common set of equipment. So, anathans, these jawless fish, actually lack RAG-mediated VDJ recombination. So they have their own system of adaptive immunity based on variable regions that are joined together and arrayed in the genome. So you remember this was the idea that this is another way that the immune system could have worked. We could just have a library of 500 pre-made antibodies and select the right one and go with it. And instead, evolution gave us this sort of toolkit, like a little box of Legos, where we can mix and match and actually tweak them a little bit to get far more combinations, like 13 million different combinations when you include the junctional diversity. Yeah. And so yeah, we will look at those. Let's have a look at these. And we're also going to mention some of these some of these genetic problems that you will see sometimes in different kinds of animals. So DSCAM is the Down syndrome cell adhesion molecule, and it's going to be an Ig-like molecule that is thought to work in insects the way an antibody does. So it's going to opsonize invading bacteria, basically. And you got your FREPs, your fibrinogen-related proteins, and these are also immunoglobulin-like. And we think they're going to do the job of innate immune system in a particular snail. Yeah, Biomphilaria glabrata. So neat. Okay, so what do these things look like? Well, here's DSCAM from Drosophila. And DSCAM is this guy, the Down syndrome cell adhesion molecule. It's the same gene that's involved in Down syndrome, one of the pathologies with Down syndrome. So it just gets this name. When we find the same thing in a fruit fly, then we call it the same name. Yeah, if we can, if it's close enough. So they've got a cluster of genes. And now, doesn't this look a little bit familiar? You've got a series of exons. So you're going to have 12 little exons of the green type, 48 of the blue type, 33 of the red type, and 2 of the orange type. And we're going to make DSCAM genes by combining together each of these parts. We got to have one from the first section, a green, a blue, a red, and an orange, basically, from the 4 different sections. And together, that'll make quite a large range, 38,000 different versions of a DSCAM protein, which is kind of neat. Okay, so yeah, so that's like a very primitive version of an immune system. Now, variable lymphocyte receptors, these are going to be things that are like on their way to becoming T cell receptors. They're going to be on the equivalent of white blood cells. And all these things become really tricky to define when we are outside of mammals. Basically, everything works a little bit differently, and knowing where to draw the line, knowing how to differentiate a T cell-like cell from a macrophage-like cell is different because they're all a little bit different and work a little bit differently than they do in people. So there's a lot of shades of gray here. But yeah, there are variable lymphocyte receptors. And yeah, so let's see these. They're going to be on the lamprey, which is one of those jawless anathan fish. And cytidine deaminase activity is, an activity caused by genes of the apobec family. We actually use this to fight against HIV, but it also, these family of genes also have a role in rearrangement of variable lymphocyte receptor genes. So this is probably some antiviral machinery that we got via the immune system of our primitive ancestors, which is really cool. Okay, so how's it work in a little jawless fish? Well, you're going to start out with a bunch of different parts, just like before. And so this is the germline conformation. So this is the DNA that our little jawless fish is born with. And it's got all the things, but they're not in the right order. They're not together. And so first thing, we're going to do a little bit of recombination. We're going to end up moving one part around and joining them together. And we're going to eventually make a complete VLR gene. And we're going to express this, and we're going to end up with all these little parts in a row and attached to a membrane. This is the little stalk part down here at the end. And so, yeah, it's a white blood cell equivalent receptor that the lamprey can make from a library of these and put together on its own. And it can not just make it, it can also make a soluble version by essentially chopping off that stalk. And so this is going to be the equivalent of an antibody, and this is going to be the equivalent of a T cell receptor. And in the lamprey, they are still basically the same gene. So this is part of what is evidence of the way that this evolutionary process actually works. Okay. Now, RAG-dependent adaptive immunity, which is what we have, is going to be based on, recombining all these different immunoglobulin-like T cell receptors and immunoglobulin-like antibodies. Yeah, immunoglobulin-like immunoglobulins. Yeah. Okay, so here's a couple little vocabulary words to help us through this section. So a DNA transposon is, a little genetic element that is able to cut itself out of DNA, copy itself, insert itself into DNA, and basically move around. They're like little jumping genes that can move around inside of your genes. And I think I remember reading this somewhere like 1 in 20 births involves another copy of this. So it involves one of these transposons either moving or duplicating itself and proliferating. A lot of our junk DNA are copies of these DNA transposons, because once they get in there, there's no good way to get them out, and they just keep moving around and, basically making babies. I don't know if you've read or heard of Richard Dawkins' The Selfish Gene. It's kind of an explanation for evolution based on genes essentially fighting it out at the genetic level. Yeah. And DNA transposons are an extreme example of very selfish genes. They contribute nothing to the host, but they keep replicating themselves, and that's why they exist, essentially. Interesting book. Yeah, got a lot of interesting stuff in it if you want to have a look at that. Okay, a transposase is an enzyme that cuts DNA, and so that's the thing that a DNA transposon is going to encode. And that's how they're able to chop themselves out and move around in the genome. A transib is a superfamily of these transposon-like sequences that we can find via a computer just by comparing bits of DNA and saying, oh, this looks like a transposon. It's 90% similar, 80% similar, something like that. And yeah, you can find these things. They don't always still work because some of them are really old and have picked up mutations that break them. But you can find them, and you will sometimes have the same transposon-like sequence, the same transib at the same place in species that have diverged 500 million years ago or more. So that's pretty cool. Yeah. And then let's see, we've got these anathen paired receptors resembling antigen receptors. These are going to be another immunoglobulin-like domain that's present in hagfish and lamprey, or all the anathens that have been properly sequenced. And these are probably, yeah, where we get our antigen receptors, our antibodies and our T-cell receptors. And they're going to be made using RAG1 and RAG2. So here we go. It's a little small maybe. But yeah, in these jawless fishes, or sorry, cartilaginous fishes, rather, this is where we get the RAG1 and RAG2. This seems to have appeared in this group. Now, there are several groups that we know of from the fossil record that sit in between jawless fish and things like sharks, cartilaginous fish. But they're all extinct now, and so we don't really know which one of these guys picked up RAG1 genes, and you know whether they had an immune system that's more like sharks and us, or whether they had an immune system that's more like agnathan fish, yeah, jawless fish. So that's a little mystery there, but they are definitely present in sharks, and so we don't know if sharks were the first to get them, or if it was in some ancestor that eventually becomes sharks. All right, and it's going to work essentially the same way. We're going to take, we're going to have RAG1 and RAG2. They're going to get together. They're going to join together little pieces of DNA. And in doing this, they're going to actually bring V and J regions together and join them up. So having this allows animals to do VDJC recombination, basically. Okay. A little bit, just to finish this section off, a little bit more vocabulary. So the bursa of Fabricius is going to be, yeah, the lymphoid organ in the gut that is the site of B cell development in chickens. And so B cells are called B cells because we're actually referring to chickens. They have this thing called the bursa, which is where all the B cells hang out. And this is different in other animals, but because the terminology came from chickens, and then we found similar cells with similar properties in mice and humans, we still call them B cells. So yeah, it's kind of neat. Even though there's no bursa of Fabricius for our B cells to go and live in. They have to hang out in the spleen instead and in lymph nodes. Okay, activation-induced cytidine deaminase is called the AID protein. And this is an enzyme that is going to cause somatic cell hypermutation. It's just going to put in mutations at random and in a way that will be permanent. It's actually going to edit the DNA. And so, yeah. If you lose AID activity, if you have a genetic defect in your AID enzyme, it's going to cause a lack of hypermutation. And you're not going to have the same range of antibodies as other people. It'll be a sort of a mild immunopathology, but not as bad as like a SCID, severe combined immunodeficient condition. Okay, a purinic and a pyrimidinic endonuclease number 1 is going to be a DNA repair enzyme. And so that's going to be involved in this class switching. Class switching is when we switch from IgM to IG, or rather M and D, because those are made together, to IgG 1, 2, 3, 4, or E or A. And IGNAR right here is the immunoglobulin new antigen receptor. And it's a form of heavy chain only immunoglobulin molecule, which is the one that sharks can make. And so this is like the big version of one of those nanobodies that the camelids, like alpacas, are able to make, and llamas. Yeah. All right. So, how's this done in different animals? Well, similarly. So we're still going to have RAG-mediated recombination because these are chordate animals and vertebrate animals, rather, that are higher up the tree than those jawless fish. And so in chickens, for example, we're going to have these VDJ and C regions or VJ and C regions, along with some pseudogenes from their old days. So these may be genes that were active in their ancestors, which were probably dinosaurs. So you can actually see what dinosaur T cell receptors and antibody equivalents might have looked like from these pseudogenes, which is kind of neat. And so, yeah, there are going to be multiple rounds of gene conversion, and we're going to end up with different affinities basically by the same process, by VDJ recombination and then picking up different C ends. So you can kind of see the way that we're set up. We've got many, many copies of our V regions together, many copies of our D regions and J regions together and in general. Although we have regions of the genome where they are organized differently. And so in sharks, it's always located with one little set together. And so they can swap out from a couple of different diversity regions, but yeah, there's only a pair of them at most, and sometimes only one. The other one's probably turned into a pseudogene and been basically knocked out. And so limited choices, but yeah, we can actually put them together. In terms of the light chains, yeah, the sharks are going to put them together, but they're going to have like a heavy and rather a variable and the constant region together, and they will then join these two things up. And so it's like a little version. It's not as powerful as the VDJC recombination that we get in humans or in mice. And yeah, it's really weird and wonderful that you can kind of see all the steps in evolution still preserved and still working to some extent in keeping chickens alive or sharks and rays, things like that. Okay, and last little bit of the chapter. So MHC class 1 and class 2 molecules are also going to be found, like equivalents of those in cartilaginous fish, which you're going to think sharks and rays and skates and things like that. But just anything that would show up in Shark Week, basically. And so you're going to have classical MHC class 1 genes. These are going to be like the ones that are in humans. And they're going to work for T cell recognition. And you're also going to have non-classical MHC class 1b genes. These are going to be also encoded, but are going to present only a restricted set of antigens. So they are like an additional MHC that just does a couple extra things, not quite as powerful as the original MHC class 1. These are going to bind, yeah, just particular common antigens that things like these sharks apparently keep running into in evolutionary terms. All right, we made it. Oh my gosh. You want to see a beautiful video on VDJ recombination? I totally recommend this. You should definitely open this up and watch it. Just when you copy it, watch out. It's going to try and put some junk between this A and this B there, and they should be both together in order to make the correct URL. That's it. And those two sections, the arthropod immune system, we're not going to cover. But just showing you that this is, we're drawing from a common set of genes. And so this idea of an immune system by recombining different parts from a library is very, very old. And it seems to exist in basically all of life. Yeah, all of animal life anyway. So that's kind of neat. All right, thanks very much. That's the end of chapter 5. We did it. We did it.